Today, we're going to look at some of the exciting new flavors of cut and tag that have translational relevance. First, I'm going to review the traditional cut and tag workflow, and then I'll get into some of the newly introduced cut and tag methods. These new methods are single cell cut and tag and spatial cut and tag. Now, both of these have the potential to add valuable detail to translational studies with patient samples. Stephen Henikoff's lab first introduced cut and tag as a low cell alternative to chip in 2019 in Nature Communications. The key takeaway from this paper is that cut and tag is an improvement upon histone-marked ChIP-seq. It's a native assay that doesn't require cross-linking or sonication, and it has a higher resolution than ChIP-seq. Cut and tag also provides better data at a lower cost with less starting material and less sequencing depth than ChIP-seq. Now let's take a look at the cut and tag workflow. First, cells are immobilized onto concanavalin A beads. Next, they're permeabilized and incubated with a primary antibody against a histone-marked target. After binding a secondary antibody, we add a protein A conjugated adapter-loaded TN5 transposase enzyme, which binds to the antibody complex. The TN5 enzyme then cuts and tags with adapters that DNA that is bound by the primary antibody, creating a library in situ. The barcoded fragments are then amplified and sequenced to a read depth of 8 million paired end reads. Now let's look at some of the data from cut and tag compared to chip. The red sequencing tracks at the top are H3K27 trimethyl peaks. At the very top track, we have the positive control, that is ChIP-seq. Henikoff's team used a read depth of 50 million. For the next three tracks, the authors changed the read depth to 8 million to match the specs of cut and tag. And this is a comparison of the performance of ChIP-seq versus cut and run and cut and tag. So looking at the cut and tag track, which is the fourth one down, we see less background here than in the 50 million read depth ChIP-seq reaction, and a slightly better resolution than the cut and tag reaction. The authors also used a cut and tag to measure H3K4 monomethyl and K4 dimethyl, as well as RNA Pol2. The pink track at the bottom is a tag-seq, and this is a good negative control. It shows that the TN5 enzyme is properly directed to the antibodies in cut and tag, and that it simply doesn't create an open chromatin profile like it would in an attack-seq experiment. Next, we're going to look at single cell cut and tag. So two papers in the same issue introduced single cell cut and tag in April of last year, 2021. Bartsovich et al. From the Karolinska Institute and Wu et al., which is Stephen Henikoff's group. Next, we're going to look at the single cell cut and tag workflow, which starts with traditional cut and tag. Like we looked at before with cut and tag, the difference, there is a difference here though, nuclei are immobilized, not single cells. The nuclei are permeabilized and incubated with a primary antibody, followed by incubation with a secondary antibody, and that protein A conjugated adapter-loaded TN5 transposase. So thus far, we have a traditional cut and tag experiment, and we have TN5 transposase cutting and tagging that DNA that's bound by the primary antibody. Next, those tagmented nuclei are loaded onto the 10x Genomics Chromium controller. This is a microfluidic device that allows for cut and tag nuclei to be met with 10x barcoded gel beads. So one nucleus meets one gel bead, and cut and tag libraries in each nucleus are uniquely barcoded. Paired end sequencing is then performed to a read depth of 250 million reads. Next, let's look at some of the data from these papers. Henikoff's group shows that major cell types in peripheral blood mononuclear cells can be identified using single-cell cut and tag. In this experiment, they performed single-cell H3K27 trimethyl cut and tag. Now, this is a common modification that represses functional elements. And shown here is a UMAP plot that clusters different cell types in informatic space. And so these cell types are defined by the common loci that are marked with H3K27 trimethyl. And they can be called out into different cell types within the blood. Here, Henikoff's group labels monocytes, T cells, B cells, and two populations of T and NK cells. Another application of single-cell cut and tag, it can identify distinct populations during embryonic stem cell development. In this UMAP plot, we can see the different stages of stem cell differentiation as defined by common loci that are marked by H3K27 trimethyl. Another figure from the same paper shows that glioblastoma cells can be identified using single-cell cut and tag. Shown here are UMAP plots that call out specific factors that are activated by having low H3K27 trimethyl in their promoters. The top left panel shows that SOX2 is aberrantly escaping repression in tumor cells. And those tumor cells are in red in the top left panel. Also escaping repression in normal neurons is RBFOX3, and in microglia is PTPRC. In oligodendrocytes, MOBP escapes the repression. So we can use single-cell cut and tag to call out tumor cells using those markers. From the Karolinska group, we can see how single-cell cut and tag can identify stages of neuron development within a population. So these, once again, are UMAP plots, but this is single-cell H3K4 trimethyl cut and tag. It shows how a single-cell cut and tag data set can complement an existing single-cell RNA-seq data set, which are the UMAP plots below. The scores are calculated by aggregating the H3K4 trimethyl cut and tag signals in the 200 most expressed genes in those corresponding RNA-seq clusters that are shown below each cut and tag UMAP plot. So for example, on the left, where oligodendrocyte progenitor cells are called out as this orange population, we can see the heterogeneity of those H3K4 trimethyl loci in the cut and tag experiment. So what this shows is that single-cell cut and tag reveals even more heterogeneity within a population than what was previously defined by single-cell RNA-seq. Next, we'll look at spatial cut and tag. That same group from Karolinska that published on single cell cut and tag has recently collaborated with a group at Yale and now presents spatial cut and tag. And this work was published in February of 2022. The key points of this article is that spatial cut and tag can regionally map histone modifications on frozen tissue sections. Here they use 50 micron squares, and we call them 50 micron pixels. So this avoids the need for tissue dissociation and preserves the geographic location of each cut and tag library. We can also derive a single cell view from the individual libraries in each pixel. And the clinical applications for this is that we can get maps of biopsies and patient tumor samples. So now let's take a look at the workflow. So if we start with a slide of a tissue section, a cut and tag is performed on that slide. We start with primary antibody binding and then secondary antibody binding. And then, like traditional cut and tag, we incubate with a PA-TN5 transposase. Next, barcodes are flowed across the slide by microchannel guided delivery in both vertical and horizontal directions. And this creates a two-dimensional grid of 50 micron squares that we can call cut and tag pixels. Each pixel has a unique combination of barcodes. So after the libraries are sequenced, we can go back and build a spatial map to see which pixels had common histone modification profiles. Now let's look at some of the results from spatial cut and tag. Shown on the left panel is an H&E stain section of embryonic day 11 mouse embryo. These authors performed spatial cut and tag using H3K27 trimethyl, H3K4 trimethyl, and H3K27 acetyl. Now, taking the cut and tag data from each pixel, they use Cell Ranger and Seurat to define clusters of cells with common signatures, and those are the different colors we see here. But each of these is the same map that is in the H&E stained slide on the left. You can also look at this data in UMAP space, just like single cell cut and tag. So this is what is the single cell view. With spatial cut and tag, you have the option of seeing the data with the pixels on the slide, or we can see the data as if we had a suspension cell solution. So the clinical application here is that it's now possible to use this method on patient tumor samples to generate epigenetic disease signatures while maintaining the geography of the tissue. So this, what we're seeing is actually the same data from the slides, but looked at in bioinformatics space rather than physical space on the slide. So in summary, single cell cut and tag identifies cell types within heterogeneous populations as defined by common histone modification. Spatial cut and tag provides a solution for frozen tissue sections while preserving the geography of the cells within that tissue. We can also get a single cell cut and tag like data from a spatial cut and tag experiment. And in conclusion, these new methods can identify disease cells within a population, making single cell cut and tag and spatial cut and tag ideal for patient samples like blood or tumor tissue. Thank you for your attention. If you have questions, you can contact me at mgarbati@activemotif.com. And for additional information, please visit our website. Thank you.